Hello everyone, this is Steven and I'm going to talk to you about using turmeric extract in treating HL60 cells. So the HL60 cells is a human leukemia cells that are mostly used in cancer research. These cells are progenitor myeloid cells which differentiate to regular myeloid cells. However, the HL60 cells are trapped in the myeloid progenitor phase and keep dividing instead of differentiating into a granulocyte or monocyte. This results in acute myeloid leukemia. Therefore, the goal of this study is to stop unregulated division of undifferentiated myeloid cells through apoptosis or programmed cell death using plant secondary metabolites that affect apoptosis. The plant extract used for this research was obtained from turmeric, which is a spice. To be able to perform this experiment more efficiently, we had to perform an in vitro experiment. Also, a special media known as IMDM, which contains a mixture of salts, amino acids, nutrients, and buffers that allow cells to grow outside of their host, was used. The media was also supplemented by 10 to 20% fetal bovine serum, which provides additional growth factors and proteins that help cells to thrive. Also, an incubator and sterile conditions were necessary for the success of the experiment. The HL60 cells were treated under a laminar flow hood, and the hood was always sprayed with 70% ethanol before working to minimize the introduction of microorganisms into the work area. The dyes used in this experiment were cytocysteine, which is a DNA and RNA binding agent, and oxidative stress detection agent, which yields a green fluorescent product when reacting with reactive oxygen species. So the first assay that was performed was the viability assay. So the main reason for doing the viability assay is to determine if the turmeric extract has any component that affect the HL60 cells growth. And this was done by adding a WST8 substrate to the cells. Every cell has an NADH dehydrogenase. So by adding the WST8, the NADH dehydrogenase will reduce the substrate to a different form, which will then change the color to orange. This is an important step because only viable cells can reduce the substrate, which is WST8, to change color. Therefore, this tells us whether the cells are viable or not. The chart here shows that the average absorbance for the turmeric is way higher than that of the control. This chart is way higher than that of the control and the cells treated with 70% ethanol. This is because the turmeric extract is already orange and hence interfered with the absorbance reading. Therefore, the chart didn't really tell me whether the cells were viable or not, so I had to view the cells under the microscope, and it can be clearly seen that there are viable cells after the treatment. There are some viable cells after the treatment. So it confirms that the turmeric has components that affect HL60 cells. The next assay that was performed was the morphology assay. The purpose of this assay was to determine if the turmeric extract was inducing apoptosis in HL60 cells by using cytocysteine, which allows us to visualize nuclear condensation and blebbing. For this assay, there were 3 controls, which were the untreated cells, solvent only, that is 70% ethanol treated cells, and camptothecin treated cells. Camptothecin is known to induce apoptosis in HL60 cells So it served as the positive control for this assay. 20 microliters of the polyphenol extract was added to the duplicate wells and incubated for 24 hours. This was then viewed under the fluorescence microscope to see the condensation, fragmentation, and the apoptotic bodies that were formed. So in my case, I did 36-hour morphology and 60-hour morphology. But before we go to that, here diagrams A and B are showing the processes or the stages of apoptosis. So from the normal cell, the cell then condenses and it fragments, start to break apart, and then finally it forms the apoptotic bodies. But these apoptotic bodies form still have its cell membrane intact, so it doesn't result in the spillage of the cell content into the body, unlike necrosis does. So in this one, we can see that the cells here, they have begun condensing, and they will eventually go through the fragmentation process and then through the apoptotic bodies. But for the 60-hour morphology, it is quite uncertain because we have some solid components from the turmeric extract that is interfering with the actual cells that we viewed under the microscope. But then we can still see that there are some cells that have begun the condensation process and about to start with the fragmentation. So some cells like these ones are actual cells that are going through apoptosis. But then the rest, we can't actually say whether they are all regular cells or normal cells that are dying, or they are just solid components from the turmeric extract. So this is actually interfering with the view, and therefore we can state with certainty that all of this are going through apoptosis. The last assay that was performed was the COMET assay. The purpose of this assay is to allow us to examine the DNA of the damaged HL60 cells. In the previous assays, we analyzed whether the turmeric extract is able to reduce HL60 cells viability or trigger an apoptotic pathway after 24 hours of treatment. This assay will help us to determine if the extract contains a bioactive molecule that is capable of inducing DNA damage in the human leukemia cells. After following the lab protocol, the cells were viewed on the confocal fluorescence microscope, but unfortunately, the cells weren't clearly seen, just like the 60-hour morphology assay, because of the solid components that were also extracted from the turmeric. But the possibility that there were DNA of the damaged cells is very high, since we had some cells go through the condensation stages in the morphology assay. So my reason for choosing turmeric. Turmeric has been used for its healing properties for centuries. For instance, it's used in a paste to heal wounds. But modern scientists have recently turned their attention to the spice, therefore releasing more publications on turmeric's health benefits. And one of the exciting areas of research is the spice's role in cancer prevention. It has been suggested that turmeric can prevent the development and slow the growth of certain cancers. So the reason why I chose it is to experiment whether it can also kill cancer cells by apoptosis. Since the orange color of the turmeric and WSTA substrate were the same, and also some solid components were extracted from the turmeric, I wasn't able to firmly confirm whether or not the extract is an inducer of apoptosis on the HL60 cells, of which I strongly believe it does. The future direction is to use a nonpolar solvent like chloroform to extract the turmeric. In this case, there will be no orange color from the extract, and also there will be no solid component to interfere with the WSTA substrate. And if this is done, it will confirm whether turmeric can kill HLCT cells via apoptosis. And this is the list of my references for this study. Thank you guys for taking the time to listen and to watch this. Have a good day.